name is Angela, and I will be discussing the premise of my ongoing PCB4943 pilot research project, the impact of caffeine on neurotransmitter production by Lactobacillus species. To begin, caffeine is a widely used psychoactive drug and is highly prevalent in our society. Due to its high consumption in many people's daily lives, I wanted to investigate the effects of caffeine on the gut-brain axis. The gut-brain axis is a biological relationship concerning the gut microbiota's effects on our brain and central nervous system. I specifically aim to determine caffeine's effects on Lactobacillus, a key bacterial species present in both our gut and in many fermented foods like yogurt. I selected Lactobacillus, specifically L. rhamnosus, L. plantarum, and L. reuteri, due to these bacteria's ability to synthesize neurotransmitters like gamma-aminobutyric acid, GABA for short, and serotonin-related metabolites. It is these neurotransmitters that will serve as my dependent variable. Currently, there is not any literature concerning caffeine's effect specifically on human gut microbiota's neurotransmitter production. However, there are existing studies that evidence caffeine encourages biodiversity and bioviability of certain gut microbiota species. Based off of these previous studies, I hypothesized that caffeine will positively impact Lactobacillus viability and, in a dose-dependent manner, positively impact Lactobacillus neurotransmitter production. However, at high concentrations, I believe caffeine will begin to exert toxic effects to Lactobacillus and neurotransmitter production will fall accordingly. My methods involve prep work first. MRS broth will serve as the media in which the bacteria will be cultured in. I also prepare glutamic acid and tryptophan to supplement my broth when I will be using it. These supplements are necessary as they are precursors for GABA and serotonin intermediate production by the bacteria. My caffeine stock solution is to also be prepared. I then plan to thaw my bacterial stock and let it culture in the prepped MRS broth overnight in anaerobic conditions. This is my starter culture in case anything goes wrong in my experimental samples. Then I will aliquot a small sample into fresh media and begin to regularly measure optical density of the sample about every 1 to 2 hours until the spectrometer has a reading representative of the logarithmic growth phase of the bacteria. I will then prepare triplicate tubes composed of MRS supplemented broth and caffeine in varying concentrations to begin caffeine treatment. The caffeine concentrations tested are 0 millimolar, which will serve as the control, 0.1 millimolar, 1 millimolar, 5, 10, and 20 millimolar. 20 millimolar is where I believe toxic effects will be observed. Each tube will have equal amounts of MRS broth and varying amounts of caffeine/saline according to the targeted caffeine molar concentration. All 18 tubes will then be incubated again in anaerobic conditions at 37 degrees Celsius for 1 to 2 days. Data collection involves getting final optical density readings and extracting supernatants for performing ELISA or HPLC to measure neurotransmitter production. With this research, I hope to contribute further insight into the growing research field focusing on the gut-brain axis. Since there is a lack of knowledge in my project's objective, I hope to contribute towards filling that gap and perhaps provide insight into how dietary caffeine affects our mood via modulation of GABA and serotonin. However, my project does have its limitations in that it cannot perfectly be generalized to a human gut. This is because I will be working with the bacteria alone, not in conjunction with gut epithelial cells. Skills that my project incorporates that were learned in my class involve using a pH meter, autoclaving, pipetting skills, knowledge of ELISA kits and chromatography, and understanding spectrometer functionality. Pilot has been the foundation for the technical skills utilized in this project and has encouraged me to look outside the box for how daily actions could impact us in ways we don't realize. It is this curiosity I hope to carry into my future career as a healthcare professional. Patients are nuanced and complex, and often daily habits could be underestimated in their holistic effects. That concludes the discussion of my project. Thank you for your time and attention.